good afternoon everyone. Welcome to the third Chemical Biology Organic Chemistry Colloquium of this term. So we're lucky to have Anna Ballard here today with us. So I think Anna's history is that she's gradually moved south. So I think you started out at Durham for an undergraduate and then did a PhD with David Smith at York. And then went across to Leeds for a first postdoc with Andy Wilson, and then a second postdoc down in London at Imperial with Ed Tate, before starting her independent career, so with a junior research fellowship at Imperial, and now she's this welcome Henry Dale fellow for the next few years. And today we're going to hear about approaches to protein-protein interaction inhibition.
Thanks very much, Sam, and thank you for the invitation. It's really nice to be here. So what I'm going to tell you about is some of the work that was done in my group as part of my junior research fellowship, and then move on to tell you about some of the stuff that's a bit more recent that I've been doing as part of my Wellcome Trust fellowship, and kind of really give you an idea of where we are now. So there's a lot of unfinished stories here, but I thought it's kind of nice to see how stuff evolves a little bit over time. So broadly what we're trying to do in my group is develop new and interesting methods to target protein-protein interactions. We're using a combination of the established methods of helix mimetics and staple peptides to target new and interesting biology that hasn't been explored previously. But then we're also working now on developing some derivatives of these scaffolds to enhance their functionality to allow us to investigate more biology with that. I'm not going to talk about that aspect today. What I'm going to focus on is our work to develop new helix mimetic scaffolds, and then talk about our work on staple peptides targeting some bacterial biology. So, starting with the helix mimetics story, what we were trying to do is to develop this class of molecules to make them a bit more synthetically accessible, basically. And that was to make a series of molecules that can target protein-protein interactions. So, protein-protein interactions are of critical biological importance. They govern almost all biological function, so cell division, DNA replication, cell death, but also determine the survival of pathogens such as malaria parasites, bacteria, and all forms of life, really. So they're inherently interesting as potential drug targets, but they are also very challenging because they tend to be characterized by very large interacting interfaces, and these tend to be quite topologically monotonous in that there are not many big binding grooves for you to design small molecules to fill. There's the key observation, though, that about 2/3 of protein-protein interactions have an alpha helical secondary structure at the binding interface, can give a structural handle from which we can start to design inhibitors. And there are two classical methods that people have used over the years. The first being staple peptides, where we can introduce unnatural amino acids onto the non-binding face of a peptide to conformationally constrain that into a helical conformation, which is its binding conformation. What I'm going to talk about first, though, would be this helix mimetic strategy, which is a completely non-peptidic scaffold, where we're really thinking about this helical peptide as like a rod-shaped structure which projects binding groups at particular spatial orientations, and then we're making rod-shaped molecules to project binding groups at those same spatial orientations and mimic that native helix. So if we look at the structure of an alpha helix first, then we have a pitch of about 5.5 angstroms, and this projects side chains along the helix at roughly every 100 degrees if you look down that helix. And that gives you side chains roughly where we've got I plus 3 or 4, and I plus 7 forming the same interacting face. And because we've got side chains every 100 degrees, that gives you roughly 3 interacting faces on the helix. A very classical example that's been very well targeted with helix mimetics is the protein-protein interaction that exists between the protein HDO2 and the helical domain of p53. And this interacts with these three key amino acids here, and a number of scaffolds have been developed over the years. This one from the Hamilton group, displaying very similar side chains which match the spatial orientation of this alpha helix. This was then expanded and developed in the Wilson group to get this oligoamide scaffold, and you can see in both cases we can get comparable affinities to that of the native peptide with this approach. But if you look in the protein data bank, what you can find is that actually about a third of protein-protein interactions are mediated via binding with more than one helical face. And when you look at some of the structures, they come from various different areas of biology. You can get some of these which are completely buried, alpha helices. These are obviously a little bit more challenging to target than that sort of surface-exposed one that I showed on the previous slide. But nonetheless, there would be significant interest in targeting these from a drug discovery point of view. So a number of groups have developed scaffolds to target interactions that occur over multiple faces, and these are some examples of the work from those groups. But I would say what really ties these together is quite long, complex synthetic routes, multiple purification steps, and just generally very challenging synthesis, which means that it's very hard for you to build up a library of compounds in order to be able to screen things, look at SAR, and that kind of thing. And because I don't come from a particularly strong synthetic background, what I wanted to try to do was to make some methods to be able to make these kind of scaffolds much more easily using pretty robust and straightforward chemistry. So what we decided to do was to combine two previously developed scaffolds where we've got side chains on this exocyclic oxygen and on the nitrogen of our amide that links this oligoamide scaffold together. And we combined these onto the same scaffold for the first time. We did some very crude molecular modelling initially to look at an extended structure of this kind of scaffold, and we can see that the gaps between the side chains that we have on a helix match quite well with what we observe on our model structure. So this was quite a nice validation of the scaffold design. Then you can carry out kind of the world's most straightforward retrosynthetic analysis of this molecule to get down to these monomer building blocks. So these are our kind of target molecules. And we can make these in just 4 synthetic steps, all of which are pretty straightforward. So we can first alkylate this 3-fluorophore-nitrobenzoic acid to introduce the first side chain, then unmask this nitro group via hydrogenation, introduce the second side chain through reductive amination, and then finally we FMOC protect to make this analogous to an amino acid. Using this synthetic route, we can incorporate most of the functionality that you find in natural amino acids, and of course we can sample the kind of vast synthetic space that nature doesn't sample itself. We can make molecules where we've got aliphatic side chains, aromatic at both positions. These are very straightforward. We can make examples where we've got polar functionality on either the O or the N alkylated position, and then we can make these double polar analogues as well. And because we've got this FMOC protection strategy and this free carboxylic acid, we can actually use an automated peptide synthesizer to assemble these into the helix mimetic structures that we saw before. So we take our monomer, which is analogous to a primary sequence, and we can build these into trimeric structures where we've got side chains mimicking 2 helical faces. And as I said, we do this in a very similar manner to peptide synthesis. We have to use an acid chloride because we've got a fairly poor nucleophilic position here, and I'll come on to ways that we're thinking about getting around that later. And as I said, using cycles of coupling and FMOC protection, we can very easily assemble these kind of structures. We've developed methods to assemble these directly on resin, which people haven't done before. Previously, people have assembled these onto amino acids themselves, but we've been able to make a fully mimetic scaffold, if you like, where we can vary the C-terminus to be either an amide or a carboxylic acid, depending on the resin that we use. So with this kind of synthetic method in hand, what we needed to be able to investigate was whether these compounds are able to effectively mimic an alpha helix. Because actually, if we look at the literature with these tertiary amides, and you look at the Cambridge Crystal Database, you can see that the majority of them exist in this kind of folded conformation. So, and that's not really a kind of mimetic conformation, and we're looking for these kind of extended modes, really. So in order to investigate conformational characteristics of these scaffolds, we made 3 representative dimers, where we're varying the side chains to either do bulky aromatic, short aliphatic, or branched, to see whether the sterics of the side chain have any impact on the conformation of these compounds. And what we're really looking at here is 3 key bond rotations. So we've got the amide bond geometry, which is kind of the central amide in this dimer, which can either exist in this kind of folded conformation, which is cis with respect to the aromatic rings, but formerly trans. So we tend to call it folded and extended to be a bit clearer with this. But this kind of bond rotation opens up to give us our extended helicomimetic conformation. We can have rotation about the aryl-carbonyl bond or the aryl-nitrogen bond, and that roughly corresponds to interconversion of helical faces, if you think about that, in terms of where our side chains are projected. So to look at this, initially we first solved the molecular structure of one of our dimers, and we find that it is in this folded conformation, which is not wholly unexpected. We can prove that this conformation exists in solution by NOESY NMR, and because we see correlation between these 2 protons and then the H2, which is these 2 positions here, we can see that on the NMR timescale we've got rapid rotation about the aryl-carbonyl bond. So the rotational barrier for that bond seems to be pretty low. What's interesting with this is we do see that we crystallize out these 2 different structures. These are rotational isomers. And this is indicative of restricted rotation about this aryl-nitrogen bond. And using methods developed by Jonathan Claydon, we were able to look at the barrier of rotation for this bond and the kind of effective chirality that you see when you lower the temperature of your NMR, where you kind of experimentally trap out these 2 signals, where they become magnetically inequivalent at low temperature, where you've got less rotation, and they gradually coalesce into one signal. And what we see with our dimers is that we have magnetic inequivalence for these 3 side chain positions, but not for this one. We were a little bit confused about that initially, but actually it's quite straightforward in that the kind of translation of this chirality, by the time you get to this position, it's just too distal from that axis of chirality for you to observe anything in the NMR spectra any longer. We see exactly the same thing for all of the model dimers, that we see this magnetic inequivalence. And when we calculate the barrier of rotation, it comes out to be about 14 kilocal per mole in all cases. So that indicates that the identity of the side chain doesn't influence the rotational barrier about this bond. That still doesn't really answer the question of whether these compounds are able to adopt a mimetic conformation or not. So for that, we turned to some DFT calculations. So a collaborator at Imperial College, Charles Romain, did these calculations for us based on previously published methods. And he looked at each of those bond rotations individually. So starting with the aryl-carbonyl bond, which we observe rapid rotation for by NMR, we can see that in all transitions the barrier to rotation is pretty low, so that correlates with what we observe experimentally. In terms of the amide bond geometry, which is really the bond that we're interested in, if we rotate around the amide bond only, then we get relatively low energy barrier, whether the side chains are on the same or opposite faces. When we look at the aryl-nitrogen bond geometry, which is the one that we see the restricted rotation about experimentally, it's quite an interesting result. if we look at a rotation around only that bond, that has the highest energy barrier that was calculated. So that gives us a barrier of almost 17 kilocalorie per mole. But if we do a concerted amide and aryl N bond rotation, we do get a lower energy barrier. So this suggests that we are at least sampling that extended conformation in solution because we are observing rotation about this bond by NMR. So whilst it's not likely to be a high percentage of those conformations, the fact that we are able to adopt that conformation for a certain fraction of time at room temperature suggests that in the presence of a protein binding partner, for example, we would be able to adopt a binding conformation. And the enantiomer interconversion that we see between the two crystallographic forms, we get very good alignment with the calculated energy barrier for that and what we've observed experimentally by NMR. So at the moment, what we've done beyond that is we've taken one of the conformations from the extended form, and we can overlay that with an alpha helix and look at the kind of average distance that we see between the side chains and what we have from the amino acids on an alpha helix, and we get a reasonably good overlay. What's quite interesting is that actually if you take our more stable folded conformation and overlay that with the beta term, we also get pretty reasonable side chain overlap. So it suggests that that conformational flexibility might be an advantage that we could tap into. So at the moment, what we're looking to potentially do is to apply some of these scaffolds to target some more challenging buried protein-protein interactions. We've looked at one so far and haven't had much success. It's quite a challenging thing to do, even with these kind of scaffolds. So we're still kind of looking to try and find the right interaction to test these against. We have also been doing some work to develop new single-faced scaffolds, because I mentioned earlier that the chemistry around the formation of that amide bond is particularly challenging. We're reacting a pretty poor nucleophile with a pretty poor electrophile. We've also got this issue of the cis-trans isomerization of the amide bond. And because we have to use pretty forcing conditions, so we use microwave irradiation at 60 degrees to form these bonds, it does restrict us a little bit in terms of what side chains we could incorporate. So what we thought to do was to change that amide for an ester linkage. So very kind of simple conceptual change. Because of the kind of anomeric effect, you get a preferred S-cis, so the extended conformation would be preferred in this case. Because of the effective anomeric effect, because of the sterics that you would see in the A12 strain that you would have in the folded conformation, and also because of the dipole moments. So we would expect this to adopt an extended conformation more favourably than the amide. So again, we've broken this down into monomer synthesis, where we start with this diphenol and we selectively protect the para position with allyl bromide. We can then install the side chain using either a bromide or an alcohol with Mitsunobu conditions, and then we can deprotect this methyl ester to give us our building block. We've successfully synthesized 10 different monomers, bearing a variety of different side chains, so aliphatic, aromatic, polar, and also charged. And I've got a master student at the moment who's trying to finish this work off and complete our series to introduce some of the more challenging side chains, so arginine can be a bit more difficult, threonine analog, tyrosine even, and then histidine and tryptophan. These are particularly challenging because with this kind of chemistry, you can get elimination when you deprotect these side chains, so we're introducing an additional carbon there to try to get around that. We've constructed some model dimers. We're pretty confident that we'll be able to adapt this chemistry to solid phase synthesis so that we can automate it in the same way, but we haven't done that yet. We've just made some dimer in solution, but we can very easily deprotect that allyl group and then couple it with a methyl ester methyl acid deprotected monomer to make these kind of dimers. So far we've just done some quite crude conformational analysis where we've done the variable temperature NMR, and what we see really is that there's not much change in the shifts of these in terms of they retain a single signal at all temperatures. So that suggests that we've either got very rapid bond rotation at all the temperatures we've looked at, or probably more likely we've got a single stable conformation. And what we're hoping is that it will be this extended one that I've drawn out here. Well, everybody said to me when I first designed this scaffold, and I actually had to take it out of a grant application because everybody was so adamant that it wouldn't work, is that these are just going to fall apart. Because esters are very susceptible to hydrolysis in water and by esterase enzymes. So with our model dimer, we had to look at that pretty early on. And we measured that in buffer at a range of pHs from strongly acidic to strongly basic. And we measured that by LCMS. And we were really pleasantly surprised to see that over a period of 7 days, across all of these pHs, we see pretty much no change in the amount of dimer that we have. We only see degradation of very high pH. So that means that the dimer is stable over very long periods of time at physiological pH, but also at gastric acid pH conditions, which is actually, we weren't expecting this level of stability, so it's really nice to see. We want to look at this a bit more, look at the serum stability and the microsome stability of these compounds, but we think it's actually quite promising. So again, that's really where we are with that scaffold. We want to try to automate its synthesis and investigate the stability a little bit more, and then we should be able to apply this to targeting some interesting protein-protein interactions. So I'm going to move on to tell you about the work that's really formed the foundation of my current fellowship, my Wellcome Trust grant, where we're using a more peptide-based approach, where we're trying to develop methods to mimic protein-protein interactions in persistent bacteria. So bacterial persistence is a mechanism by which bacteria can survive environmental stress, which can include antibiotic treatment. So in response to treatment by antibiotic or any environmental stress, the population will generate a small subpopulation of a dormant phenotypic variant known as a persister. And the problem with this is that antibiotics often target the molecular mechanisms which govern rapid replication that you see in your normal bacteria. So these persisters are capable of surviving quite extensive antibiotic stress. And importantly, these bacteria can revert back into a normal phenotypic variant and re-establish the original population. And this is not only a problem in itself, given that it can often be the underlying cause of recurrent infection, a particular problem for diseases like typhoid and tuberculosis, but this also necessitates repeat courses of antibiotics to clear essentially the same infection, which can again contribute to treatment failure through the development of genetic resistance. So of course the reason that we're particularly interested in that is because persister formation is governed by protein-protein interactions, particularly those that occur between toxin proteins and their natural inhibitor proteins, the antitoxins. So in response to environmental stress, the antitoxin is degraded to release the toxin to allow it to arrest bacterial growth and initiate that dormant state. So what we want to try to do is to make something that which mimics the activity of that antitoxin to try to reverse or prevent persister formation. So at the moment we're looking at Salmonella, which has 14 different toxin-antitoxin pairs, and our collaborators, Sophia Lane's group, did a series of genetic knockouts to look at the effect of each toxin-antitoxin pairs on the number of macrophage-induced persisters. And you can see that this pair that exists between DOC and PhD has the biggest effect on persister numbers compared to the wild type. So DOC is a fictamine kinase, which phosphorylates an elongation factor, EFTU, which inhibits protein translation in order to initiate that dormant state. It's inhibited by the C-terminal alpha helical domain of the antitoxin PhD. So, as I said, what we want to try to do is to make some effective mimics of this antitoxin protein to try to reverse persistent formation. So we're not trying to inhibit this interaction, more replicate one that's no longer there. In order to do this, of course, we need to have access to all of the proteins that are involved in this interaction. We can express and purify the antitoxin very easily and also the substrate of DOCK EFTU. But in general, expressing and purifying toxin proteins is very, very challenging. If you overexpress them, particularly if we're looking at salmonella proteins, which we overexpress in E. coli because they're very similar bacteria. So overexpressing the toxin effectively over-inhibits your bacterial growth by targeting the EFTU that's in E. coli. And you get these kind of growth curves, which don't give you usable amounts of bacteria. If you mutate out the catalytic histidine, we can restore normal growth. But in this case, the majority of the protein that we're isolating is completely insoluble. One of the strategies that we tried was to introduce an exporter sequence to shuttle that protein out into the periplasmic space which exists between the cellular barriers in gram-negative bacteria to try to remove it from the substrate protein. That didn't really help at all with the toxic expression, so we still see exactly the same growth curves. And again, that modified toxin protein was very insoluble. So the strategy that we came up with, I think is quite neat. What we're doing is exploiting the natural mechanism of DOCK to get around this. So what we can do is co-overexpress the toxin DOCK with a mutated EFTU that cannot be phosphorylated. So we've knocked out the phosphorylation site, a threonine, replaced it with an alanine or a valine. So compared to DOCK alone, or if you overexpress with wild-type EFTU, you see toxic expression. But when we overexpress either of our mutant proteins, we can get back to normal growth. And that means that compared to overexpressing wild-type EFTU or none at all, we start to get significant overexpression of our toxin. We can measure the phosphorylation of EFTU by Western blot very easily. And this means that we can start to isolate at least usable amounts of this protein. So this method has still required some optimization. So this would be your kind of normal protocol, if you like, where we generate bacteria cells which encode the mutant EFTU, and then we would transform with a plasmid that encodes for a histac toxin DOCK. Normally you would then plate this out on an antibiotic resistance plate, select colonies, inoculate in a small overnight culture, and then take some of this and scale it up. That doesn't work for us because we do still have a problem in that we do have a bit of, even when we haven't induced the expression of DOCK, there's a little bit of leaky expression, which means that we can get some toxicity. So we've removed a few steps. We've removed the plating out steps, so we transformed and immediately inoculated with an overnight culture. That doesn't really work very well for overnight expression. So what we've got down to now is basically the minimum protocol that we can possibly get away with, where we take our chemically competent cells expressing mutant EFTU, transform, and immediately put it in a large-scale expression culture. So it's quite nice because it's much more efficient. We're getting through a lot more cells this way because we have to inoculate with more of them, but it does mean that we can start to isolate usable amounts of protein. So the next slide is one that, that my postdoc Guillaume made for me to show his purification protocol. So he's purifying these on a Talon column to trap out the histag. We can then run this down a size exclusion column to isolate monomeric DOCK. And then you've got your pure histag DOCK sample. And when we concentrate this, we have about 2.5 milligrams from 2 litres, which is significantly better than what we had before. But the problem is a few seconds after, a lot of this protein crashes out, and we end up with between 0.5 milligram and 1 milligram per protocol, which is a little bit soul-destroying, I think. And what this comes down to, really, is the stability of the toxin protein. In the conditions that we were using here, we've got a melting temperature of about 33, 35 degrees. So it's not a very stable protein. We've actually, in the last few weeks, been optimizing this a lot. We've done a massive buffer screen, and we've been able to shift this to about 55 just by changing components of the buffer. So that should mean that we can start to at least preserve more of the protein that we isolate, which means that we'll have enough to do things like crystallography and ITC. But at least with the amount of protein that we've been able to isolate, we've been able to set up some binding assays. So we've made a fluorescently labeled PhD peptide, and we titrate that against the concentration gradient of the toxin to attempt to calculate the KD. We're not confident in the accuracy of this number, which I'll come on to. But we can then use this in a competition mode using the same measurements, where we're competing out with an unlabeled analogue of the fluorescently labeled peptide. And this we can measure IC50s and use this as a screening tool to compare different DOCK inhibitors for their affinity. And we get a very similar profile when we use our 23 amino acid peptide versus the entire antitoxin protein. What's particularly nice about isolating active protein is that we can look at the phosphorylation of the FTU as a readout for the activity. And we can do this by Western blot, and we can see with the full recombinant PhD protein that we inhibit phosphorylation at higher amounts. And we're really pleased to see that we get exactly the same effect when we use just our peptide. So that's a really nice validation of our strategy to just target that antitoxin binding domain. So I mentioned that we weren't totally happy with the KD value that we're measuring. And that's because the kind of hint here is the steepness of this slope. So this should be a 1-to-1 interaction, so the slope for this should be about 1, but it's more like 2. Also, if we vary the concentration of our fluorescently labeled peptide, we get a variation in the KD value that we calculate, which is also not what you should see. And this is because what we're actually dealing with is an incredibly tight protein-protein interaction, possibly immeasurably tight. So it's always worth reminding ourselves that we aren't trying to inhibit it, we're just trying to mimic it, because we think this is going to be sub-nanomolar affinity. So initially we were using fluorescein labeled peptide, and we can get down to about half nanomolar of that concentration, but we're still getting this same issue with the Hill slope. And at this point you're really limited by the brightness of your fluorophore. So we've swapped recently to using TAMRO, and here we're starting to maybe get towards the values that might be realistic. KD, when we start to get down to these tracer concentrations, the KD isn't varying too much, and we're getting a much more sensible slope on that graph. But you can see from the kind of error that we're getting with this fitting that we're, again, pretty much at the detection limit for our plate reader. We're getting some new filters next week to try to kind of open up the brightness for that, because the TAMRO filters you tend to buy attenuated, so it's effectively like they come with sunglasses on, because the fluorophore is bright, and normally people want to avoid that. So I'm going to try with a new filter, which should mean that we can get down to even lower concentrations and accurately measure the KD. And also, once we've got more protein, we should be able to back this up with ITC, and we're going to start some SPR measurements this week. I'm slightly worried about the kinetics of this interaction, whether the off rate is going to be incredibly slow, but we'll see. What we can do though with this assay is still use this to measure IC50s to gain some information about the molecular detail of this interaction. So we've done this using an alanine scan to try to investigate this. We're working on a homology model. We're working on Salmonella typhimurium interaction. The published crystal structure is from a different bacteria, Bacteriophage P1. So we built a homology model for our interaction. But it's important to look at each residue to kind of back up that homology model, but also to tell us a bit about the individual binding contributions of each amino acid. So we've made an alanine scan library where we're substituting out each of the amino acid side chains, and we can divide these roughly into two categories, where we've got those that point towards the binding interface, and then we have the solvent-exposed residues, which we wouldn't expect to have a contribution to the binding. And as expected, when we replace each of those, we don't see a significant change in the IC50. And that's also true for some of the interface residues. So these at the end terminus of the sequence that we're looking at don't have any effect on binding, and that's quite nicely backed up by some of the data in bacteria that Sophie's group have got, where they can truncate this peptide a little bit and still see the same reversal. But we wouldn't have necessarily expected that these two residues wouldn't have much effect on the binding affinity. The remainder, though, of the interface residues, so these 6 amino acids, when you mutate out one of those, you get an order of magnitude, if not 2, decrease in binding affinity by IC50. So these are what we would classify as the hotspots of this interaction, and the ones where we would maybe struggle to mutate these out without impacting the binding. We've started to look a little bit about the contribution of each of these individual helices, because this is quite interesting kinked structure. We've made fluorescent analogues of either the C-terminal or N-terminal helix, and what we saw was complete obliteration of all binding up to very high concentrations, which is something I wasn't particularly expecting. So we think that there's likely to be the case that we need to kind of fill this whole binding pocket with something, or that perhaps binding of one end sort of templates the binding of the other. We've done some multiple hotspot deletions to look at that a little bit, so we've deleted both the hotspots from the C-terminus, and there we do see binding, so it does suggest that there might be some kind of templation effect, but we're going to look at that by SPR to look at kind of saturating one helix and then titrating in the second one to see if we can pull apart the importance of each one. Of course, if we want to target bacterial interaction, it's really important that these peptides can get inside. That's very challenging for gram-negative bacteria in particular. So with our fluorescently labelled wild-type peptide, we did some uptake experiments with microscopy and flow cytometry, and what you can see here is pretty much nothing, which shows you that there's absolutely nothing getting inside the bacteria. But because we've done our hotspot analysis, we now know which amino acids we can substitute out. So we can replace some internal amino acids for arginine, and that increases our uptake so that we can at least measure it. So we can quantify this by flow cytometry, and we get almost nothing with the wild type, and that increases to about 11% when we introduce these arginines, which I think is not bad for a first go. We've of course looked at the binding affinity of these mutated peptides, and unfortunately it does seem like if we replace that many arginines, we do have an impact on the binding. So the second generation of peptides we've made is a more classical approach where we've introduced the same amount of arginines onto the N-terminus of the peptide, and that doesn't have any impact on our binding. So we're hopeful that we'll be able to replicate that same improvement in uptake without that impact on binding affinity. But one of the other things that we're looking to do is to generate staple peptides of these sequences, because we know that that has been shown to improve cell uptake. And that's because if you're using peptides in a biologically relevant environment, there are fundamental issues with doing so. Removed from the stabilization of an entire protein, a peptide adopts millions and millions of random coil conformations, and very rarely would choose to adopt this helical fold in solution. When it's unfolded, it's also significantly more susceptible to enzymatic degradation, and peptides tend to exhibit pretty poor cellular uptake, which I've just demonstrated for you. So one of the things that we can do, which I mentioned right at the start of the talk, is this technique called peptide stapling, where in this case we can introduce 2 unnatural amino acids with this alkene functionality, and then use Grubbs metathesis to covalently cross-link these together and constrain this into a permanent alpha helical conformation. And this was shown last year to work in improving the uptake in gram-negative bacteria. So we're hoping that we'll see similar effects with our peptides. So last year I had a BSc student, and at Imperial they do just a 4-week project in the lab. So I'm pretty impressed that he managed to make 3 different staple peptides from this. where we're substituting out these surface exposed residues at positions I and I+4 from each other because we know that they don't affect binding, and that should be a reasonable position to put the staple in. And then we did the kind of classical experiments that you do with these peptides where we looked at their helicity by circular dichorism, and we can see that that does vary a little bit depending on where we put the staple. This is potentially because we've got that kinked helix structure. And then we've looked at their stability, just their stability to trypsin degradation by LCMS. And in just 1 hour, our unstable peptide is completely degraded, whereas the stapled analog is stable, which is what we would expect. But of course, we need to look at this in much more robust degradation conditions to kind of fully validate that. And then we can show that this doesn't seem to have an impact on the binding. At this point, it is difficult for us to measure whether we're getting an improvement in binding affinity because of the limits of the assay that I discussed before, but it seems like we're not losing any binding affinity by doing so, which is a really good thing. So we're going to do a bit more investigation in terms of different types of stapling chemistry, complete the set to look at all of the positions that we could look at, and then look at their uptake into bacteria to see if we get any improvement. So that's really where we are with that project. So today I started by telling you about the work that we did during my junior research fellowship, where we developed 2 new helix mimetic scaffolds, one double-face and one single-face, based on straightforward chemistry capable of automation. We've done quite extensive conformational characterization of our double-face scaffold to look at its ability to adopt an extended mimetic conformation in solution. And we've shown that our ester scaffold is completely stable in physiologically relevant conditions. So with the Persister project, we've developed a method to express and purify a very challenging toxin protein, and we've characterized the hotspots of that protein-protein interaction for the first time. And we started to develop some preliminary stable peptides, which seem to improve their stability. So it just remains for me to thank the people that have been involved in this work. So this is my current group members. My postdoc, Gilliam, has been doing a lot of work on optimizing the protein expression protocol that was developed initially by Fiona Rowan. My PhD student, who finished in February, Theo Flack, did pretty much all of the helix mimetic work, alongside Mohammed Haque, who during his master's project worked with Theo on the ester scaffold. Anna Lasada de la Lastra did all of the alanine scanning work during her master's project, and Pete Gobbett was the BSc student who managed to turn out 3 staple peptides in his 4-week project. Very grateful to my collaborators Charles Romain for the DFT and Sophia Lane, who's a Persister expert, formerly at Imperial, but she's recently moved to Harvard. And these guys for the facilities at Imperial, and then these organisations for funding. Finally, you for your attention. Thank you.
Okay, thank you very much, Emma. That's perfectly on time, and a great breadth of science from chemistry all the way through to biology. Pete, I have a question, I think.
As it happens, Sam, there might be a possibility. So, for a lot of helices, when they're interacting with things, in addition to the side chain stuff, there's the helix dipole that can be quite important. Now, with the polyaryl amide and the polyester, presumably there is little or no dipole similar to the helix dipole. Is that true? And has anyone even thought about it? I bet no one's even.
We haven't particularly thought about that, I'd say.
So very often you find substrate sat at the top of a helix, which has got a huge great dipole on it, and it's effectively adding up to a whole positive charge at one end of the dipole and negative charge at the other, depending on. So I just wondered whether anyone thought about it, but it's kind of irrelevant because you're doing all the side chain stuff anyway. I just thought I'd ask.
Yeah. I haven't especially thought about it, I suppose is the answer.
We'll go on from Ali, but anything from anyone further back first?
Students go first. Does anyone know?
Okay, fine.
Why do you think the think that you have to have an equally tight-binding molecule to have
I
it?
don't know. It's an interesting question because also with the melting point stuff, so last week we did some analysis with the PhD and we see a massive stabilisation upon binding, which I guess is also expected. So it might be that because they're both fairly unstable proteins, particularly the antitoxin, we would expect to be even less stable. But there's not actually a lot known about the mechanisms for reversal, exactly what triggers the antitoxin degradation, the kind of regulation of the expression of the toxin and the antitoxin is quite complex. It's a kind of conditional cooperativity-based mechanism. So actually, a lot of these things we don't really know. So I think, aside from potential therapeutic benefits down the line, I think having chemical tools to explore that is going to help quite a lot. So I think, if we could show something that binds in vitro, whether that will be enough in bacteria or not, I guess remains to be seen. Yeah.
Anyone? Peter?
So PBAD vectors are slightly less leaky than PET vectors, and you just induce with a rabbit host rather than.
So the EFTUs are the PBAD, because we've got them under two different.
Okay. Don't know if that. So you might want. So you might have to put them as or do it as an operon. So put the two genes in the same thing and express together.
Yeah.
And then you won't get the leakiness, and then at least your first phase of growth will be healthy. It'll be horribly toxic once you switch it on, but it's going to be horribly toxic. Right?
Yeah.
Just an idea. Sorry
Yeah.
about that.
Okay, anything else? Matt?
In the first part of your talk, when you're talking about conformational analysis of your peptidomimetics, which conditions which solvent basically?
So by NMR, we've done
In
it.
NMR,
We've done it in chloroform, DMSO, we've done it in DMSO water mixtures as well.
Okay, so because I was thinking, and maybe it's very naive, rational, assuming two major possible conformations, the overall difference in energy in water and in DMSO may be different, right?
Yeah.
Because of the intrinsic properties of the solvent.
Yeah.
Do you observe anything like this in your experiment, depending on
So we
which
observe the
solvent you're
same
going
barrier of rotation irrelevant of the solvent that we've used.
Okay. I'm thinking about things like burial of hydrophobic surface and how
So
it
we haven't
bounds
done
to-
the calculations were done in DMSO, I believe. But we haven't looked at multiple solvents in the calculations.
because they may be more constrained in water,
Yeah.
including salts and things like this,
Yeah.
than in DMSO.
Yeah.
But
I think-
DMSO solubilizes everything almost.
Yeah, but I say so by NMR at least we didn't see any major differences when we titrated in water.
Okay.
They're not soluble enough, unfortunately, to do it in 100% D2O.
5%?
Yeah, we went up to like 30% water, so that's kind of as much as we can push it. Because we're cooling it down as well, then
Crazy.
yeah.
Thank you.
Okay, quick question
Yeah.
about synthesis, solid phase one. So obviously the temperature is very powerful. Everything that we've done has always been in solution phase. And how long can you make these things? And what kind of efficiency do you get?
So we didn't go beyond making trimers with the double-faced scaffolds, but previously in Andy Wilson's group they made a decamer with a single-sided one. So I can't see much reason why we couldn't make something that. I think you start to be limited by the solubility of your monomers more than anything else.
But you think potentially you could make one of the ones with your...
Yeah, I'm pretty sure. I think what will be potentially nice with the ester one, because the chemistry is a bit milder, we'll be able to do that at room temperature. Then we can potentially use a parallel synthesizer, which works at room temperature, and make 24 in one go. So that's the kind of idea with that one, rather than using a microwave one, which makes them one at a time. But so with that one, we're going to try, because it's new, chemistry.
Yep.
We're going to try and see how long we can make it with those as well.
Cool. Okay, if you don't have any other questions for Anna, thank you for a great sport.